Hello, welcome to the Life Sciences Core Laboratories. I am Dr. Pflügel, Director of the Life Sciences Core Laboratories and your instructor for the laboratory course LS23L. All of the lectures are administered online, therefore you will probably not meet me in person. You will be taught by your TA during the weekly 3-hour laboratory sections. However, if you have any questions or concerns, I am happy to meet with you in person, and you can always contact me via following email drpflugel@gmail.com. The following presentation will cover some background needed for your first laboratory. But before I cover the specific information regarding your first lab, I would like to go over some general information regarding this laboratory course curriculum. The course has its own web portal, and some of the labs will use some of the links provided there. Included here are also links to the weekly lectures and lab instructional videos. Furthermore, this portal provides general information about our lab personnel and specific information as the weekly section schedule, the quarter schedule, the syllabus, and the week-by-week assignment sheet. For instance, there is an abbreviated to-do list available via the weekly link, which contains the assignments for each lab and deadlines. For example, to prepare for the pre-lab and in-lab quizzes, you have to read the pertaining sections in the lab manual and watch the online lectures. Now, I want to focus on some key aspects of this course as mentioned in the syllabus. You will complete 10 different laboratory sections over the course of the quarter. The lab will cover topics in physiology and molecular biology. If you not have had LS3 yet, or are not currently enrolled in LS3, which is our molecular biology lecture course, or if you do not have any equivalent course credit, you should not be enrolled in LS23L. Some of the labs are heavy on molecular biology, and you will not be able to master the laboratory and final materials without prior knowledge of LS3. LS23L is also a lab course with a heavy scientific writing component. You will be writing 3 larger scientific style papers. The 3 research papers are spaced out during the quarter. Labs A in week 1, Lab D in week 4, and Lab G in week 7 will require you to summarize your experiment in a scientific type research paper. The papers are due in lab the following week, so you have 1 week to finish the paper. Please check the dates to make sure you make time to complete the papers. The papers are due at the beginning of your lab section in weeks 2, 5, and 8. Make sure you look at your schedule and economize your time accordingly. There is a late submission penalty if you do not submit your paper on time. Besides the 3 larger written research papers, which you have to complete at home, you will complete 7 in-lab worksheets. Here's an overview of the workload. You will complete 10 laboratories in 10 weeks, including 10 online lectures, 9 pre-lab online quizzes, 1 lab safety quiz before Lab 2 in week 2, 9 in-class quizzes at beginning of lab, 7 in-class worksheets, and 3 larger scientific papers. We will finish the quarter with 1 multiple choice final in week 11, which will be scheduled individually online later in the quarter. Therefore, each week you will have to attend 3 hours of laboratory, and before lab you will need to listen to online lectures, complete online assignments, and complete online pre-lab quizzes. This will take about an hour to total per lab. Please make sure you have enough time to complete all these assignments. There will be 10 online lectures available to you through CCLE and our lab website. Most lectures are structured in the following way. The presentation will be divided into roughly 3 parts. The first part presents an overview of skills and concepts of the lab. The second part presents some background about the lab. This might be information as delivered in our LS2 or LS3 lecture courses. If you are firm in the knowledge of these courses, you could skip this information and go right to the third part. The third part will go over some details of the lab you will be doing. It will also provide information on lab safety. This will conclude my introduction to the laboratory course LS23L. I will now provide some information about your first lab. What follows will be the first part of the lecture. For your first laboratory, the CMIT, or the Computerized Memory Interference Test, this lecture will give you some background concerning the lab you are about to perform, and will introduce you to an ongoing research project here at UCLA in collaboration with Cedars-Sinai. The lecture is about 30 minutes and needs to be completed before you come to lab. The second part of this week's lecture will go over guidelines on how to write a proper lab report. This lecture needs to be completed after you have done the lab and before you're about to write up your laboratory. For your first lab, please go to the Science Learning Center, which is located on the 4th floor in Young Hall South. The room information is available online, either under the Room tab or the Section tab. Once you come to lab, there will be two parts to this lab. In the first part, you will watch the instructional video, read the IRB, and then perform an in-lab quiz on CCLE. Then you will perform the neuropsychological measure, the CMIT. In the second part of the lab, you will formulate a hypothesis and then test it using the CMIT database. The laboratory skills you will meet in this lab include calculating a p-value using a t-value and table, using a web database to retrieve demographic data, using PubMed or other academic databases to retrieve a peer review article. The concepts covered in this lab will include understanding the concept of a null hypothesis, understanding statistical analysis, understanding rejecting or accepting a null hypothesis by interpreting a p-value, and understanding the structure of a scientific paper. I will now invite Dr. Enrique Lopez to present his research lecture about neuropsychology and the memory interference test to you.
Hello, my name is Dr. Enrique Lopez. I'm a clinical research neuropsychologist. I'm an assistant professor at Cedars-Sinai Medical Center, and I'm also an assistant clinical professor at the David Geffen School of Medicine at UCLA. In the next few slides, I'm going to introduce you to neuropsychology. I will also provide you with the history and background of the Computerized Memory Interference Test, the CMIT, and its relevance in neuroscience research. I hope you enjoy the lecture. What is neuropsychology? Neuropsychology studies the relationship between brain and behavior. We know that different brain areas are involved in different types of behavior, so by measuring behavior, one can determine how different brain areas are functioning. This can be very helpful to determine if an individual suffers from a particular neurological condition. A neuropsychologist evaluation is conducted by a clinical psychologist with postdoctoral training. Neuropsychologists are involved in many neuroscience research and can help make important medical decisions. A comprehensive neuropsychological evaluation usually involves a full day of testing. Later on, I will give you the opportunity for you to experience a short measure that can be useful in detecting attention deficit hyperactivity disorder. This measure is specifically designed to assess impulsivity and inhibition. Let me begin by explaining some basic functional neuroanatomy. This cartoon design is a representation of the behaviors associated with the two sides of the brain. On the left side of the brain is usually where language, logic, analytical thinking, math, science, and language are located. This is for about 90% of the population, and usually associated with the opposite of one's dominant-handedness. The right side of the brain expresses abilities in spatial, creativity, spirituality, imagination, and identification of colors. Now let's divide the brain from a different perspective, from the front and the back part. The frontal part of the brain is usually associated with abstract reasoning, problem solving, decision making, attention and concentration, and behavioral control, while the motor cortex is responsible for movement, while the sensory cortex is where one experiences body sensations. The parietal lobe is responsible for perception and making sense of the world, while the occipital lobe is responsible for vision. The temporal lobe is where hearing and language and its understanding is processed. As you can see here, the entire body is expressed in both the motor and sensory cortex. And as you can see, the body is represented upside down with the legs way on the top of the brain and the face at the bottom. It's also important to note that the motor and sensory cortex is contralateral, meaning that the right side of the brain controls the left side of the body. One side of the brain controls the opposite side of the body. Now let's look at the brain from a different perspective, from outer regions to the inner regions of the brain. In the outer areas, which are called the higher cortical areas, these tend to be related to cognition, language, and visual and attention abilities, while the more inner areas or subcortical areas are typically responsible for basic survival needs, such as wakefulness, thirst, and breathing. Sometimes when individuals suffer from a traumatic brain injury and it results in a coma, it's usually because they actually damaged some of the more inner areas, in this case, the brainstem. So how do neuropsychologists measure cognition? Actually, most of the measures are paper and pencil types of tests. None of the tests hurt, and they merely involve answering questions. However, some neuropsychological tests may be challenging for anyone taking them. Tests are usually standardized with published age, education, and gender norms, which assesses the area of cognitive functioning. However, we still have to obtain better cross-cultural norms and develop more culturally fair tests. Now let me demonstrate a sample cognitive test. This test was developed by Stroop and has 3 components. In the first session, you're going to read words. What you're going to do here is you're going to read these words down in a column. We'll start with red. Then you're going to go down to green, blue, red, and so on. You're going to go down the column, and then you're going to start back up in the next column, which indicates blue. And then you're going to go down the second column, and you'll keep doing that until I tell you to stop. When I tell you to stop, you'll be presented with a page like this that will demonstrate how many words you have read. If you understand what you're to do now, let's proceed. Okay, now this page, now let's see how far you can get by reading the words as quickly as possible. Are you ready? Set, go. Now, try to remember the number of words that you were able to read. The next task that you have to complete is name the colors of the ink as fast as you can. You're going to go down in columns like you did the reading task. You'll begin with blue, then red, green, green, and so on until I tell you to stop. Are you ready? Set, go. Now try to remember the number of colors you were able to name. Okay, now this task is a little bit more difficult because you have to name the color of the ink, like you just did in the previous example, while ignoring the written word. For example, this first item over here, the ink is blue, the next one is red, green, green, and so on. Keep naming the colors of the ink while you ignore the written word as fast as you can. If you make an error, correct yourself. Continue down the columns until I tell you to stop. Are you ready? Remember to name the color of the ink, not read the written word. Are you ready? Set, go. Okay, try to remember the number where you left off. As you can see, this condition is much harder. Most individuals find the last condition the most difficult, meaning that they produce the fewest items in the word color interference. So if you find yourself having difficulty with the last condition, it doesn't mean you have any cognitive problems. The comparisons would be done with a standardized version administered to a normative data sample of individuals with similar demographic characteristics such as age, education, and language. We know that the fastest is usually word reading, and that's because it has been overlearned, and it's all on one side of the brain, which is usually on the left side of the brain, while color naming is on the opposite side of the brain. In order to speak out the color, it has to come back to the left side, and it takes a few milliseconds more to do that process. The color word interference is the most difficult because the brain has to make a decision utilizing the frontal lobes. It has to choose to do the task that is most difficult and not the one that is overlearned. Therefore, it has to inhibit what is easier. Now let me explain what likely happens in the brain under the three different conditions of the Stroop. Let's begin with the first task where you are required to read a word. First, the occipital come into play because it has to see the word, and then it goes to Wernicke's area, number 2, because you have to understand what you are actually reading, and then it goes to number 3, Broca's area, where you actually are going to say the word. The fourth section is where your lips are going to say the word out loud. As you can see, reading typically is all done on one side of the brain. Now let's go over the brain area related to naming colors. In this area, you're going to do the same thing in the beginning, where you begin in the occipital lobes. However, this time your brain has to go to the other side of the brain where your nonverbal color identified abilities are. Then it has to be processed back to the Wernicke's area and then to Broca's area so that you say the word out loud through your lips. As you can see, the color naming should take a little more time. Now let's talk about the brain areas related to inhibiting the behavior, and that's the last condition, the color word interference condition of the Stroop, the most difficult task. In this task, you're required to use the frontal lobe. The frontal lobes is used to make decisions, and you're required to name the color and not the word. So it tells you to do what's more difficult and not what's easiest. It tells the brain to go to the other side of the brain so it can identify the color and to come back and continue with the process to name it out loud. As you can see, the frontal lobes is the decision mechanism that helps regulate and suppress impulses. One thing that is very interesting is to note is that the frontal lobes are usually not completely developed at the age of 25. That's actually why maybe car injuries are high when a person is under the age of 25. And that is why the insurances are higher, because people may not be able to inhibit responses and behaviors or make better decisions. This may actually help you also understand why a stimulant may be helped an individual who may have an impulsivity disorder. It actually requires a lot of energy to concentrate and focus and inhibit behavior. It requires more energy for a child who is angry to inhibit hitting another child because the frontal lobes are necessary for inhibiting responses. Such individuals are not getting enough oxygen or stimulation in the frontal lobes to be able to focus and concentrate. What I like about the Stroop is that it actually can help diagnose individuals who have ADHD without causing any judgment in someone's behavior. The Stroop can help explain why someone may be having difficulty inhibiting behavior through a simple naming and color naming task. Hopefully, with the demonstration of the Stroop, you were able to see how these neuropsychological measures can be very useful in detecting neurological conditions. As you can see, the Stroop is a very short measure, but it can provide us with valuable information. A comprehensive neuropsychological evaluation evaluates many different domains. For example, intellectual functioning, attention, abilities such as reading, writing, and math, language, visual spatial abilities, memory, motor abilities, and decision-making abilities. I will now introduce you to a measure that assesses memory. Now I'm going to provide you with a history of the picture memory interference test. The World Health Organization in the late 1980s launched a multi-center study to explore the nature of cognitive disorders due to HIV in different regions of the world. We do know that the HIV virus crosses the blood-brain barrier, and a significant number of individuals infected with HIV suffer from neurocognitive disorders. The World Health Organization was concerned that a lot of individuals throughout the world may suffer from undiagnosed neurocognitive disorders, so they developed several cross-cultural measures, and one of those measures they developed was a picture memory interference test. The original study was conducted in different parts of the world, such as Brazil, Germany, United States, and Kenya. In the United States, the study was conducted in Los Angeles. This is the first publication of the PMIT in the British Journal of Psychiatry in 1991, along with the other neuropsych measures that were used. A more updated version of the picture memory interference test was created through the use of technology, and why it's now the computerized memory interference test, the CMIT. With the use of technology, the test is much easier to administer because in the past it was administered using flashcards that were presented in 2-second intervals. We also added alternative stimuli to remember, such as audio, reading words, faces, designs, and kanji. Additionally, we are able to explore additional research questions, such as what would happen if we presented the stimuli at 1, 2, and 3-second intervals. We also were able to measure how long it takes an individual to make a decision, measuring reaction time. Reaction time can give us information if an individual takes a long time to make a decision, or if another individual responds too quickly, but inaccurately. Over 10,000 students have participated in the CMIT, creating a significant database. The objective of the undergraduate research initiative at UCLA was to teach and expose undergraduates about research design and cross-cultural neuroscience in the life sciences core education laboratories. With such a diverse cross-cultural database of the computerized memory interference test, we may learn more about cross-cultural neuropsychology and about different cognitive disorders such as Alzheimer's, autism, and traumatic brain injuries. Additionally, other institutes across the world are utilizing the CMIT, such as Mexico, Puerto Rico, Brazil, and Korea. Presently, HIV continues to be a medical concern and to impact a variety of ethnically diverse individuals, LGBT communities, and other disenfranchised populations in the United States. We're very fortunate that the World Health Organization and UCLA initially developed the PMIT to be used with diverse populations so that we can utilize with individuals from diverse backgrounds in the United States. Let me demonstrate how the computerized memory interference test is structured. If you agree to participate anonymously, you will be asked to complete a demographic lifestyle questionnaire. Then you'll have a pre-testing trial that will help you navigate the keyboard and how to endorse your answers. Then you'll be presented with a book of pictures, book 1, that you'll have to memorize. Then soon after, you will be presented with another set of pictures, which you will indicate yes or no if you just saw those pictures. Then you're presented with a new set of pictures to remember, book 2, which you'll have to remember only those pictures, and hopefully it will not have interference with previous pictures presented in book 1. Again, you will then be presented with a new set of pictures, book 3. You'll have to only remember those pictures. As you can see, the task will become more and more difficult to inhibit pictures from the past. Book 4 will require you to remember where you first saw those pictures in book 1, 2, or 3. The last trial will be a simple reaction test where you'll have to just simply say yes or no if you see a particular picture. Below you will find a sample picture. Each book displays 20 pictures at a 2-second interval. Once a book has been presented, then you'll be presented with 50 pictures, and you will have to respond yes or no if you saw those pictures. The maximum of correct responses is 20 for each book, 1, 2, and 3. This is a complicated slide to demonstrate the different data that we have collected. For example, looking here, we can see students' first language is English, then Chinese, Korean, Vietnamese, Spanish, Persian, and Armenian, and so on, for those who took the CMIT utilizing the picture stimuli. Other valuable demographic information is available for you to explore for your lab assignment. Again, having a computerized version of the test has given the opportunity to evaluate different types of stimuli. For example, here is data presented under 3 stimuli conditions with both the numbers correct by trial and the corresponding reaction times for picture, word, and audio. This slide demonstrates some of my results of some of my research of HIV-associated neurocognitive disorders, otherwise known as HAND, on the CMIT. Right here are 2 sets of groups of individuals. Both of them are HIV positive. However, there's a group who suffers from HAND and then one that does not. We can see that the picture memory interference test on book 1, 2, and 3 looks like it distinguishes between individuals who have hand and don't have hand. You can see here on the people who don't have hand that they almost score perfectly a 20, and the test does become more difficult for everybody, and so everybody eventually drops. However, there's still a difference between the group that has hand versus the one that does not have hand. Hopefully this gives you a great introduction for this cross-cultural memory measure, and that it has sparked your interest in neuroscience research. Again, thank you for your attention, and hopefully you will enjoy the computerized memory interference test.
Thanks go to Dr. Lopez for this inspiring lecture about the neuropsychological measure, the memory interference test. I hope you all enjoyed your first online lecture. With this, I will end this lecture and wish you all a great time in lab next week.